so first of all, I want to try and connect this a little bit to Queller and Strassman's definition of organismality, or what an organism is, and it seems like your research does have a very tangible or specific way of kind of backing up that definition of what is it, high cooperation and low conflict, and the consolidated bundle of adaptations making whatever. So I'm sort of curious of like how you think that this research does enforce that definition or doesn't, and also why yeast. Like, I don't know, this is maybe a separate question, but how is it that you found yeast, or I don't know, maybe yeast is typically studied in this type of field, but I think you mentioned at the end of your talk another similar research with algae, I believe, and I'm just kind of curious about why yeast and how other things like maybe algae also support this kind of definition of what an organism is.
Yeah, no, great question. So in terms of Queller and Strassman, it's kind of interesting. Like when you wade into the philosophy on this, it seems like there's kind of there's sort of two different. Like that, I think a lot of people use the term organism and individual interchangeably, and some people have been kind of trying to pull those two concepts apart. And I think, and so the way I tend to think of it is that organisms, and this is kind of borrowing Peter Godfrey Smith's ideas, which I, of course, take Queller and Strassman and a bunch of other people's stuff kind of into the same account as well. But an organism is something which is essentially a functionally entwined entity. So like a great example that Peter Godfrey Smith likes to use is a squid, a bobtail squid, and a Vibrio fisheri, which is fluorescent and inside its lux organ, or its light organs, right? So they're an organism because they work together and they're highly cooperative and they're functionally entrained. But they are not an individual because they're not an evolutionary unit, right? So if you define something as a Darwinian individual is a participant in an evolutionary process, then the squid is an individual on its own right, and the Vibrio is an individual in its own right. But because they basically have this short-term ecological association where the squid flushes out the Vibrio every day and takes new ones into its light organs, their coevolution is pretty minimal. And basically, they're not like a lichen, which is like maybe a more persistent association of a photosymbiont and a fungus, which is both an organism and a Darwinian individual, because selection is acting on the symbiosis itself, and they're being co-inherited in propagules and stuff. Or us with mitochondria, in which we're very much part of the same individual. And so the way I tend to think of it is that organisms essentially fit the Queller-Strassman definition of high cooperation, low conflict, functional integration. Whereas individuals are essentially the way I describe things of, do you have a basically heritable variation or traits that affect fitness? Is selection acting on those? And as a result, are you seeing adaptation? So it's very much an evolutionary, as a description of the evolutionary process. And you're asking, what is the focal point of adaptation? What is the focal point of selection? And those two things aren't always, they're usually the same for the kinds of things that we see with our eyes. Trees, for animals, they're the same. But when you really kind of get down into these gray zones, they're not necessarily the same. So I think Queller and Strassman are, I think they're largely, I don't think there's anything wrong with their work, obviously. I really like that paper. But the reason that I don't tend to talk about organisms as much as individuals is because I think it's possible to have higher level individuality, say, really without necessarily cooperation or conflict playing a large role. I think usually you will have cooperation and conflict, but I don't think that that's necessarily the defining trait. I think the defining trait is the evolutionary process. And cooperation and conflict are things which arise in the context of the evolutionary process and basically make the organism what it is. But to me, that's a result of evolution. And so it's the process of how is the evolutionary process playing out. That's the fundamental thing. And so that's why I like to kind of focus on, just because like normally, so for most organisms, you can't really observe that process. You can get an idea of where adaptation, you can get an idea of the process because you can look at traits that have evolved. And if I say, look, this organism has a bunch of cells that are being altruistic, and that appears to be adapted for the group, then you're pretty sure that cells aren't the focus of selection, right? It's hard to explain it if they're a society of cellular individuals. But it's easy to explain it if the cells are a part of the multicellular individual. So, if looking at traits often gives you insight into the evolutionary process, but if you can study the process directly, then why not? Because it's rare that you can. To me, that's really the fundamental part.
So that's where the yeast comes in, right? I mean, where else can you study the evolution on that level?
Yeah, I mean, you need a model organism that you can grow under controlled circumstances, and you can know a lot about them. And one of the nice things about yeast is that you can freeze them, so I can actually go back to the actual ancestors of anything extant. It's easy to modify them genetically, so I can take genes that evolved in the future and put them into the past, or take genes that evolved in the future, repair them to what they were like in the past. You can do all sorts of great experiments with them that would be really hard in other organisms.
So yeast is basically...
Go ahead.
So yeast is basically like time travel, is what you're saying.
Yeah. Freezing yeast is time travel, for sure. You can freeze other organisms too, and we chose yeast really pragmatically. It's just an easy organism to work with. The downside of that is that the upside is there's a huge community of people studying yeast. So much is known about them. Any method that you want, you can do in yeast. Like, oh, I'm going to do CRISPR. Okay. Who's got a CRISPR system in yeast? This guy. Can I have it? Sure. It's great. It's easy. Downside is that people really expect you to do a lot of cell biology because you can. And if that's not really your main interest, then it can be kind of a drag. It can be a lot of like, oh, we would publish this in Nature, but you don't have no cell biology. And if you had an organism that didn't have as many resources, they'd be like, well, we understand why you didn't do all the cell biology. So it opens doors, but then people kind of want you to go through all those doors, even if that's not the goal of that specific project. They kind of want you to go. They want you to do all the bells and whistles for every paper, which is that's the downside.
That sounds like a state of affairs that is not particularly unique to yeast research, although I can definitely see why it'd be acute there.
It's not unique to yeast research. That's right. But if I was working on some marine, and single-celled eukaryote, which is a close relative of animals and didn't have a genome, I could get away with doing a really different set of experiments than I can get away with yeast, for example. No one's going to expect you to nail the SNP and then do single-cell transcription profiling. Because no one's ever done it before. Well, yeast, look at these guys. Their entire lab spent 4 years making this process work, so you should do that kind of thing. That's my snarky take on it. But no, overall, it's a good thing because so many things are possible, and I think that's a great thing.
Well, and your evidence is just so strong. Like, I read your first paper, and I was like, holy crap! I don't know, that you got this so reliably and so quickly. It's kind of unprecedented for artificial selection experiments.